factor is the time. Like if you are exceeding the time, then you are automatically disqualified. So please be careful on that. That is very, very important. And then, of course, you would have done all your presentation and everything. And so when I start, when I say start, and I will say that start only when, your audio, video, everything you have checked up and you find that, yes, now we can go ahead. Only when you tell that to me, I will say start. Then Dr. Renuka Devi will keep up the time for us.
Okay, madam.
Okay. And before we get into, I don't know how many of you here know Professor MJ Narasimhan. He is father of MJ Tirumalacha. And I mean, sometimes when you read whatever he has done, MJ Narasimhan, in that year, like he was born in 1891 and had this very, very peaceful demise in 1970s. And you'll be surprised to know that he advocated the biocontrol against the insect in those years. And he is the one who devised the spraying instruments, and perhaps well known for his contribution for the understanding of the involvement of Phytophthora ericae in the cholera disease of areca nut. And he worked under Professor Coleman, and who is again one of the pioneering work in those years. So it is really, it's not a question of winning or losing. It's a question of, in his name, we are only recalling whatever the scientific discoveries had been made by him. And then, improve ourselves so that, we also can emulate him and then, work like him. So in that memory only, this contest is being conducted. And I hope you all understand the spirit of this contest and then keep it up. So without much delay, I welcome Dr. Amoga. Where are you? Amoga?
Yes, sir. Yes, ma'am. Yes, ma'am.
Amoga Varsha. You can do your share screening and audio you test is, and only when all the thing is okay, I will say start. Okay? You are ready?
Just a minute, madam. Is it audible, madam?
Yeah, it's audible. It's audible to everybody?
Yes, ma'am.
Yes. So now you can mute yourself and switch it on only when the 13th minute or 15th minute comes. You are ready, Amala?
Yes, ma'am. Yes.
Okay, your time starts now.
Good afternoon, everyone. I welcome to this my presentation. Title of my research is Characterization and Virulence Profiling of Magnapurtha Orange Isolates from Diverse Rice Ecosystem of Karnataka. We all know that due to the urbanization, we have lost most of the agricultural land, and there is a conversion of agricultural land into a important cities or something else. We are losing our land. So there is also increase in the world population. Today it is about 7.8 billion, and there is increase in the demand for the food, and there is fight for the food, so that there is a need of more of production of the food grains. Major of these is the staple foods like maize, wheat and rice. And coming to the Indian context, the important staple food is rice, and also for South Indian conditions, rice is a very important staple food, and it is also known as global grain, and 42% of the total grain production. It contributes to 42% total grain production, and about 92% of the paddy is consumed in the Asia. And coming to this rice, sufficient production is not happening because of since this is cultivated in many ecosystems like irrigated upland, hills and deep water. It is affected by the majority of the important diseases like blast, fall smut, bacterial leaf blight, etc. And coming to these important diseases, the first one is rice blast. So the rice blast is seen under more than 85 countries, and around 10 to 60% of the harvest will be lost every year. Even 10% of the loss is sufficient to feed 60 million people for 1 year. And this can be managed by many of the methods, but the important one is host plant resistance, because we know that it is cost friendly and farmers prefer this resistant varieties over time. But the thing is that the involvement of human beings and environment are making the plant susceptible, and there is a breakdown of the genes over time, and hence there is need to find the resistance genes for a particular ecosystem. So with this context, we have undertaken the research with the title Characterization and Virulence Profiling of Magnoporthosis isolates from Diverse Rice Ecosystem of Karnataka. The objective of my research is survey to access the rice blast disease status and then to find the virulence spectrum of these blast isolates of different rice ecosystem, and then multi-locus sequencing of these isolates and final identification of the resistance source. I have almost covered the entire state covering growing the rice, and these are the different locations. From here I have collected the sample during my study, and I have surveyed around 110 villages of Karnataka covering 18 districts, and these are the different symptoms observed during my survey. And this is the difference between the healthy crop and severely affected field. And we have used IRRI 2013 scale for the severity assessment, and this is the scale which I have developed during my work. And these are the different ecosystems surveyed, and these are the different districts where we have observed highest disease severity in Chikmagalur district of hill ecosystem with 81.60%, whereas the least was observed in Gadag district with 1.68%. And coming to the ecosystem wise, the hill ecosystem has 53.3% disease severity, whereas the least was in coastal ecosystem with 3.37%. We have put this into a map, and we have found that wherever the rainfall is between 700 to 4,000 millimeter, the disease was severe between 20 to 60%, whereas the extreme rainfall of more than 4,000 mm in case of coastal ecosystem has also not resulted in much of the disease severity, and even the lowest rainfall of less than 700 also resulted in the least disease. And when we have analyzed with graphical representation, we have found that the ecosystems like hilly and Kaveri or some of the other irrigated ecosystem with the rainfall from 700 to 400 to 700 mm have the highest disease severity compared to the coastal and least disease areas. So we can say that the as we know that rice blast is associated with the high humidity, the incidence of the rainfall under cloudy conditions has resulted in the most severe of the disease in case of hilly ecosystems of the Karnataka. And next objective is virulence profiling of rice blast isolates. The steps involved are collection and multiplication of the isolates, establishment of the monogenic lines, preparation of the inoculum, and inoculation of the differential lines and observations. And I have used the isolates collected during my survey to this objective, and I have collected 52 isolates and named them from MOK 1901 to MOK 19052. And I isolated the Magnoportha oryzae by spore drop technique on 2% water agar and subsequently cultured on the PDA. And these are the spores which we have used for the isolation. Means after seeing under the microscope, I used a single spore isolation method for the isolation of the blast. And during the process, I also characterized this for morphological characters like color, form, elevation, and margin. And we have found that grayish color, grayish black color, grayish cottony, grayish white, and whitish cottony color isolates, and circular form isolates and irregular form isolates, and raised and flat and petiform isolates. And also the margins were entire or filiform or undulating in nature. And after that, we also use these cultures for the virulence profiling. We have schematically represented here the culturing of the pathogen. Later on, the sporulation will be induced and then inoculated on the plants, that is nils, and then it is kept for the incubation overnight in the inoculation facility and then transferred to the glasshouse for 1 week and then observed for the disease symptoms. These are different origin lines having particular. We have used 24 origin lines. One last one is a nil, that is LTH background, and all these having a particular origins, and these are obtained from the Katak, and we have sown these in the trays, and after when they have reached 14 days, we have inoculated the samples with our isolates. That is, we have let our culture to sporulate under proper conditions, and then we have kept for the inoculation facility, and then we observed for the symptoms. We can see resistance and susceptible reaction of this pathogen on the lines. And this is the representation, overall representation of the process which we have carried out, that is inoculation, culturing of the pathogen, and side by inoculation on these lines, and then observed for the disease symptoms. And this is the scale we have used for the scoring, that is 0 to 3 is considered resistant and 4 is susceptible. And this is the chart table which this is the result which are obtained for this reaction. These are the isolates showing the particular reaction on this particular origin lines. And with this, we have found a highly virulent means isolates with breaking more than 9 R genes in case of irrigated ecosystem and 10 genes in case of rainfed ecosystem. And around 10 genes were found resistant for this isolates in case of irrigated, and 4 genes for in case of rainfed ecosystem. And this is the graphical representation where we can see the percent breakdown of the R genes by particular isolates of the different ecosystem. And the last one is that is LTH, showing 100% disease breakdown by all the isolates of the collection. And during our investigation, we found that the isolates from the rainfed ecosystem are more virulent in nature compared to the irrigated ecosystem. And based on this, we also classified the isolate into 14 pathotype groups. The 14 pathotype groups in which 2 pathotype groups, that is 6 and 7, were dominant with 6 isolates each. And we also identified the effective origins for particular ecosystem. And these can be used for breeding program in case of gene pyramiding or either in alone. And these are the clusters. We have classified the isolates into 3 clusters, which are distributed at different rates at the different ecosystem. And coming to the third objective, that is multi-locus sequence analysis of the Magnaporthorace isolates. We have used 38 isolates for this study for collected during the survey, and the process involved is growing of the pathogen on PDB. The 7-dose culture was used for the DNA isolation. Later, it was using specific primers. They are amplified using PCR, and the PCR product was purified and allowed, then sent for the sequencing using Sanger sequencing method. And later, the sequence were analyzed by using BioData and MegaX software. And we have used the 5 parameters like actin, beta tubulin, calmodin, translation elongation factor 1 alpha, ITS, and we have used the particular reagents for PCR, and then we have used this thermoprofile condition. And these are the different genes amplified during the study, that is actin, beta tubulin, calmodin, translation elongation factor, and also ITS. These are the accession numbers which are obtained from the gene bank for ITS, actin, beta tubulin, and calmodin. And with this isolates sequences, we have constructed the phylogenetic tree, and it was classified into 2 clusters. The cluster 1 consists of the majority of the isolates, except except the cluster consists of only one isolate, that is Magnapur 1906, Yamoka 1906, which is from the hill ecosystem of Karnataka, showing divergence from the major cluster. And this one is the outgroup, that is Magnopurthe griseae. And our last objective was identification of the resistance source. We have screened 1,265 lines during our study, and we have used uniform blast nursery. And we have found 96 highly resistant lines, 359 resistant, 310 moderate resistant, 125 moderate susceptible, 49 susceptible lines. And these are the different host reactions we observed during the study. These are the different entries. And this is the graph showing the different reaction pattern, that is 96 were highly resistant, to 359 resistant, 310 were moderate resistant, 125 moderate susceptible, 49 were susceptible, and 326 were highly susceptible. And this is about my work. And in conclusion, among the different ecosystem, hilly and cover ecosystem recorded severe disease outbreak compared to other ecosystem of state. 52 representative isolates of Magnaporthorace are collected from each ecosystem, and significant morphological diversity considering their color, form, elevation, margin was observed. Further, to understand the virulence diversity among the eco-distinct isolate, internationally accepted pathotyping was carried out using 24 IRBL lines, and 14 pathotype groups distributed in the different rice ecosystems of Karnataka, in which pathotype group 6 and 7 were moderately virulent but predominantly distributed across the state. And the study on the virulence profiling was also reviewed the effective R genes to be used for the developing ecosystem specific resistant cultivars. And we employed the advanced molecular tools such as multi-locus sequence typing and to find the genetic relatedness among the isolates. And based on this, we have used 5 genes, that is actin, beta tubulin, calmodin, ITS, and translation elongation factor. And we have found similar kind of branching in case of actin, ITS, and TEF1. And we found that it can dissect the isolates effectively compared to beta tubulin and calmodin. And the phylogenetic study also revealed the different ancestral origin of the blast isolate from the hill ecosystem. And we have also attempted to identify the resistance source against rice blast disease using 1,265 different breeding lines and found resistant sources. Thank you, madam.
10 minutes, 55 seconds.
This is in brief about my work, and the outcome of this work is we can use this different R genes for inbreeding program to develop resistant lines, either in single or in combination.
Thank you very much, Dr. Amoga Varsha. Normally in the Narasimhan contest, we don't entertain questions. There is no question and answer session at all. Okay. Otherwise, it's very good work, and application also you have proved it, and I'm very, very happy for you. And congratulations for the good work done. Okay.
Thank you, madam. Thank you.
Okay. So You have to stop your sharing of the screen.
Yes, ma'am.
Is there any hitch that you all people had in hearing or in, there is no discussion that is very well known in MJN Award, and you cannot ask questions. So I call upon the next person, whoever is available, Dr. Cynthia,
Yes.
please come in.
Yeah.
Does anybody have any other suggestion or it's going on well? Hello?
Yes. Shall I start my presentation?
1 minute. Are you ready? You are,
Yeah.
everything is ready? Okay.
Yes.
You start then.
Okay. Good afternoon all. My presentation is on molecular characterization and functional analysis of pathogenicity related genes in Colticum falcatum causing sugarcane red rot. A brief intro about sugarcane. It's an important commercial crop grown for its diverse uses. And the crop is susceptible to several biotic and abiotic stresses in nature. Among fungal disease, red rot is of serious concern that is caused by the pathogen Colticum falcatum. And this is called as cancer of sugarcane since they reduce cane yield and sugar recovery. Coming to the background, this disease actually poses great challenge in developing resistant varieties of sugarcane since the pathogen is gaining virulence and also simultaneous breakdown of host resistance. The hemibiotropic nature of this pathogen involves a large set of pathogenicity genes that plays a major role in the development of the disease. So better understanding on these genes involved in pathogenesis would pave a way for a management of the disease through transgenic approach. And also we can identify defense related genes for the control of the disease. So based on this, the objectives are framed to characterize the pathogenicity related gene homologs in C. falcatum, then to identify the pathogenicity related genes that are differentially expressed during pathogenesis by SSH approach. And the third objective is to validate selected pathogenicity genes during interaction by qRT-PCR. And finally, to standardize and functionally analyze selected pathogenicity genes by RNA approach to agrobacterium mediated transformation. So first for this study, two isolates were chosen, that is the highly virulent isolate CF671 and least virulent isolate CF92020. And these isolates showed both genotypic and phenotypic variation among them. And coming to the first objective, that is to characterize the pathogenicity related gene homologs in C. falcatum. About 28 genes homologs were referred from other Colticum species and amplified in both the isolates of C. falcatum. And the results showed amplification in both the isolates or either in virulent or in the least virulent isolate. And this gene sequences of the gene homologs that were amplified in C. falcatum showed intraspecific variation among the isolates, and also they showed more than 90% identity with other Colticum species. So coming to the implant expression analysis, here both the isolates were inoculated in the susceptible variety COC671 and the expression was found to be varied. That is, it expressed either in the virulent on 5th and 10th day or only in the least virulent isolate. So in conclusion of this objective, about 28 genes were characterized in response to pathogen virulence. And in addition to that, in-plant expression analysis also revealed differential gene expression in response to pathogen virulence. Then second objective is like identification of pathogenicity genes that are differentially expressed during pathogenesis by SSH approach. So for this, 4 sets of SSH analysis was performed. Here you can see the sample reaction that given in the table. And first SSH was performed to identify the genes related to pathogenesis. Then second SSH to identify the genes related to C. falcatum virulence. And the third SSH was performed to identify virulence gene in response to sporulation. And finally, the fourth SSH performed to identify the genes responsible during compactable interaction. So these isolates were inoculated in the respective host, and SSH analysis was performed. And finally, the subtracted products were sent for sequencing at nucleome. So this sequence analysis, raw reads that were obtained were trimmed, and fungal clean reads were obtained, and that fungal clean reads were subjected to the Bouty tool, and finally the unigenes were obtained. And for this unigenes, pathway analysis was performed using KEGG automatic annotation software. So coming to the results of this objective, here these are the isolates, that is the sporulate virulent isolate and non-sporulating least virulent isolate and moderately virulent non-sporulating isolate. So these isolates were inoculated in the respective canes. and RNA was isolated and SSH analysis was performed. So this is the subtractor PCR products that were sent for sequencing a nucleome. And sequencing results showed about 37 unigenes were expressed. And these are the list of unigenes that were differentially expressed from all 4 sets of SSH analysis. And among 37 unigenes, about 10 were commonly expressed in all 4 sets. So the pathway analysis of the unigenes showed in first SSH about majority of the unigenes were involved in translation process, and in second SSH majority of the unigenes were involved in signal transduction and translation process. Then in third SSH analysis, largest functional category here is the carbohydrate metabolic process, whereas in fourth SSH analysis, the largest functional category is of translation process. So in conclusion, from all 4 sets, about 37 unigenes were identified, and among them, 10 were commonly expressed. And in addition to that, pathway analysis of the unigenes revealed that majority of the unigenes were involved in translation, signal transduction, and carbohydrate metabolic processes. So coming to the validation of selector pathogenicity, unigenes expressed during host pathogen interaction by QRT-PCR. Here 4 reaction sets was performed. That is, the highly virulent isolate was inoculated in the susceptible variety, then least virulent isolate inoculated in the susceptible variety individually, and then the highly virulent isolate inoculated in resistant and moderately susceptible variety. And for this study, about 20 differentially expressed unigenes were selected, and RNA was isolated from the inoculated skein samples, and QRT-PCR was performed based on the optimized conditions, and the results were shown as heat maps generated using Excel stat software. So coming to the results of this objective, the inoculated cane samples were collected on 24, 48 hours, 5th and 10th day post-inoculation, and QRT-PCR was performed. So the results showed that differential expression of the genes was observed among the isolates at varying intervals. And here the bright green color represents the upregulated one, whereas the bright red color shows the genes that are least regulated, and the rest of the genes are moderately regulated. So here also differential expression was observed. That is like some of the genes expressed in the virulent isolate during compatible interaction, whereas during incompatible interaction, their expression was downregulated. And in conclusion, validation of the unigenes resulted in differential expression at varying intervals among the isolates in response to pathogen virulence. And moreover, high expression of selected unigenes showed their significant role during pathogenesis, especially from the virulent isolate. So coming to the standardization and functional analysis of selected genes by RNA approach, here 3 genes were chosen. That is polyketide synthase, involved in melanin biosynthesis, glucose transporter, and sucrose non-fermenting gene that are involved in carbohydrate metabolic process. So in polyketide synthase, acyl transferase and conidial pigment polyketide synthase region were chosen. And for these gene, the primers were designed, and sense fragment was first amplified individually for the genes in the highly virulent isolate of C. falciparum, that is CF671, and it was introduced into the vector P71 vector, that is the fungal transformation vector in sense orientation. And then similarly, antisense fragment also amplified individually for the genes in C. falciparum isolate. Then that was introduced into the recombinant P71 vector in antisense orientation, and after that the recombinant P71 vector was introduced into the binary vector, and then the recombinant binary vector was introduced to Agrobacterium tumefaciens LBA4404. Further, the recombinant Agrobacterium was co-cultivated with a fungal spore suspension of C. falciparum isolate CF671, and finally the transformers were obtained, and that was selected with suitable markers. And for that, the mutants were subjected for cultural observation, and the meiotic stability of the mutants also observed. And in addition to that, pathogenicity testing of the mutants was performed by stock inoculation and by detached leaf bioassay method. Here in stock inoculation, the mutants were inoculated in standing cane of COC671 and isolated at varying intervals. Later, detached leaf bioassay method, third leaf of COC67 was excised, and the mutants were inoculated on the pinprick midrib regions of the leaf. Then molecular confirmation of the mutants was performed by amplifying hygromycin and target gene. And in addition to that, the mutants were analyzed by QRT-PCR for expression. So coming to the results, here you can see the target sense amplification in C. falciparum, and their insertion in the vector was confirmed by restriction digestion. And similarly, antisense fragment also amplified, and their insertion was confirmed by restriction digestion. So finally the target was released and confirmed from the pCambia, recombinant pCambia vector, that is the binary vector, and it was co-cultivated with the fungal spore suspension. And the results showed the knockdown mutants produced no spores, while the wild type isolate produced spores. And the meiotic stability of the mutants also showed that second and third generation mutants were similar to that of the first generation mutants. Then coming to the pathogenicity testing by inoculating in COC 671, the inoculated cane samples were collected at varying intervals like 24, 48 hours, 5th, 10th, and 15th and 30th day post-inoculation. And here you can see the wild type isolate showed disease development more than 2 internodes covering the entire width of the cane on 30th day, while the mutants showed disease development within the inoculated internode till 10th day, and on 15th and 30th day, the disease development was more than 1 inoculated internode, but it was like covering half the width of the cane. So all the 4 mutants also showed same results. And coming to the detached leaf biopsy, here you can see that third day the mutants exhibited no lesion development, while on 5th and 7th day, except SNF mutants, all the mutants showed reduced lesion development compared to the wild type isolate. So in confirmation, molecular confirmation, here the hygromycin gene showed strong expression, whereas the absence of the target gene was observed. And expression analysis by QRT-PCR also showed more than 70 to 80% reduction of the genes in the mutants. Then in conclusion, we standardized RNA approach for functional analysis in Coltricum falcatum, and this SNF expression pattern of the sucrose non-fermenting gene seems to alter the fungal energy source, thereby causing the disease. So coming to the output, that includes about 28 genes were characterized in C. falcatum for their in response to virulence, and from SSH analysis, 4 sets of reaction was performed that resulted in 37 unigenes, and among them 10 were commonly expressed. So in addition to that, validation of the unigenes also showed differential expression of the genes among the isolates in response to pathogen virulence. And further, RNA approach showed that was for first time standardized in Coltricum falcatum, and all these 3 genes that were knockdown mutants showed their clearly indicates their role during pathogenesis. And moreover, the expression of the hygromycin gene and absence of target gene shows a successful insertion of the vector into the fungus. And coming to the outcome, based on these genes, we can go for identifying the gene marker for a screening program in response to pathogen virulence. And the candidate genes involved in C. falcatum pathogenesis may serve as a platform to identify the defense-related genes, and can also be considered as a potential target site for developing inhibitors in control of the disease. Then based on pathogen adaptation phenomenon, genes responsible for a pathogen gain of virulence and breakdown of resistance can be identified. And besides all these, the transgenic, the knockdown genes in this study could be further taken for transgenic approach through Higgs, that is host-induced gene silencing mechanism, either alone or in combination of genes. Thank you.
Madam,
Yeah.
12 minutes, 46 seconds.
Oh, Dr. Sindhya,
Yes, ma'am.
I mean, very, very elaborately done work, and
Thank
I
you.
really congratulate you for doing it.
Thank you.
And you also have shown the future direction, how your work will be useful. And that way it is very good. I congratulate you. Okay.
Thank you.
And our next participation. Ranjana Joshi is available. Hello.
Hello, ma'am.
Ranjana Joshi is available.
Ma'am, yes, ma'am.
Yes, ma'am, she is there.
Yeah, you have to load the slide and tell me, no? I'm not able to see.
Okay, ma'am.
1 minute. Okay. Have you loaded your slides?
Ha, ma'am.
Okay, then you are ready. You can start.
Ha, ma'am. Yes, ma'am.
Please
Okay.
full screen. Go for full screen.
Go for full screen.
Am I audible, ma'am?
Yeah, you are audible. You are audible.
Little bit louder, please.
Okay, ma'am. Shall I start, ma'am?
Yeah, start.
Good afternoon, everyone. I am Ranjana Joshi from College of Agriculture, UAS Raichur, Karnataka. I am here to present for Professor MJN APS Award, and my topic is identification of bioactive compounds in native trichoderma and fluorescent pseudomonads for management of wilt of chickpea caused by Fusarium oxysporum, former species Saiceris. Since we all know that the chickpea is the world's second most important pulse crop after the common bean, and India is the major chickpea producing country, which contributes over 75% to the world chickpea production. And among India, Madhya Pradesh ranks first in area as well as in production. This is the area production productivity in India and in Karnataka. Since this chickpea is prone to many abiotic and biotic stresses, among which the soil-borne disease wilt is an important disease of chickpea, and it causes yield losses up to 100% under severe conditions. By considering the importance of the crop and importance of the disease, the following objectives were set for my research work. Before going to objectives, it is must to know the symptoms of the disease and causal agent. It starts with yellowing of the leaves, later drooping and drying were observed under field conditions. And at severe conditions, complete wilting of the plant is noticed. When we cut open this stem, there is a discoloration of vascular tissues was observed. And the causal agent is Fusarium oxysporum, former species Saiceris. This is the taxonomic position of the pathogen. Since the perfect stage is not yet known, this is the pure culture of the Fusarium oxysporum, former species Saiceris on PDA medium. It produces well separate branched mycelium and micro and macroconidia and chlamydospores intercalary and terminally. And this was subjected for pathogenicity test on variety Anigeri 1, and the pathogen has proved its pathogenicity on Anigeri 1. Coming to my first objective, that is collection of native trichoderma and fluorescent pseudomonads from healthy rhizosphere of chickpea and their antagonistic potential against Fusarium oxysporum, former species Saiceris. Since I have collected 20 different trichoderma isolates from different northeastern Karnataka districts, and they are isolated and designated from TR1 to TR20, and these were subjected for cultural characterization, in which all the 20 isolates varied in colony growth from 75 mm to 90 mm, and colony color varied from white to green. Colony shape varied from circular to irregular, and all the type of growth was fluffy and raised in all isolates, and margin varied from smooth to wavy. Here we can see the cultural variability in 20 different isolates of trichoderma. Similarly, the fluorescent pseudomonads are also collected and designated from PF1 to PF20 from different northeastern Karnataka districts. These are the pure cultures of fluorescent pseudomonads on Kingsby medium. And these 20 isolates were subjected for morphological characterization, in which all the isolates produced fluorescence under UV light, and in Gram's reaction all were negative, and the cells were rod in shape. This is the pure culture, and in Gram staining all were Gram negative, and this is the fluorescence under UV light. In biochemical characterization, all the isolates were positive for KOH test, catalyst test, gelatin liquefaction test, and starch hydrolysis test, except PF2, PF9, and PF10, which were negative for gelatin test and starch hydrolysis test. Here we can see the sting observed in KOH test and bubbles observed in catalyst test, and there is a liquefaction in the gelatin medium. And in case of starch hydrolysis, there is a zone around the colony of the bacteria. And these 20 trichoderma isolates were subjected for their antagonistic potential against the pathogen. All the isolates significantly inhibited the growth of the pathogen under dual culture technique, among which the TR14 isolate has shown maximum inhibition of the pathogen by 91.85%, followed by TR10, and least inhibition was observed in TR16 isolate. Here we can see the maximum inhibition of the pathogen by TR14 isolate. Similarly, the fluorescent pseudomonads are also tested against the pathogen, in which all the isolates have significantly inhibited the growth of the pathogen. in which the PF19 isolate have maximally inhibited the growth of the pathogen by 88.89%, followed by PF15, and least inhibition was observed in PF3. So here we can see the inhibition of the pathogen in PF19 isolate. And these 20 isolates of trichoderma further tested for volatile compounds production by inverted plate technique, in which all the 20 isolates produced a significant amount of volatile compounds which are inhibitory to the growth of the pathogen, among which TR14 has produced higher concentration of volatile compounds which inhibited the growth of the pathogen by 72.22%, followed by TR19, and least inhibition was observed in TR18. Here we can see that the volatile compounds inhibited the growth of the pathogen. In case of fluorescent pseudomonads also, PF19 have produced a higher concentration of volatile compounds, inhibited the growth of the pathogen by 66.67%, followed by PF14, and least inhibition was observed in PF9. So here we can see the PF19 has produced maximum amount of volatile compounds. One efficient isolate, that is PF19 of fluorescent pseudomonad, was identified as Pseudomonas fluorescens based on morphological and biochemical characteristics. And in case of trichoderma, the trichoderma, it was identified as Trichoderma asperellum based on PCR reaction, and the sequence was deposited in gene bank, and this accession number is obtained. This is a pure culture of Trichoderma asperellum on PDA. And this Trichoderma asperellum TR14 and Pseudomonas fluorescens PF19 were used for further studies. This is the Trichoderma asperellum produces separate well-branched mycelium, and we can see the branching of the conidiophores, and the oblong conidia will be produced on phyllates, and chlamydospores are also observed. Coming to my second objective, that is extraction and identification of bioactive compounds in efficient isolates of native Trichoderma and fluorescent pseudomonads. This is the schematic representation of extraction and identification of the bioactive compounds from Trichoderma asperellum. The extract was obtained by solvent extraction method, and that extract was fed to the GCMS MS system, where we obtained 29 different volatile compounds were observed in the extract of Trichoderma asperellum with different retention time and area, among which 2 imidazole 1-allyl methyl pyridine 1 oxide was observed at the highest area. That highest area in the chromatogram represent the higher concentration of that compound in the extract of Trichoderma asperellum. Here we can see the chromatogram of the compounds of Trichoderma asperellum, which were obtained at a different retention time and at different peaks. This is the structure and mass fragmentation of that particular compound, which was present at higher concentration. This is the structure of the compound which was present at the highest peak. Similarly, the extraction was done by the Pseudomonas fluorescens as same as Trichoderma asperellum, and the extract obtained was fed to the GCMS MS system, and we obtained 55 different bioactive compounds, that is volatile compounds, in the extract of the Pseudomonas fluorescens with a different retention time, among which the DL-leucinamide N2N2 dimethyl was obtained at higher area, which represent the higher concentration of this compound in the extract of the Pseudomonas fluorescens. Here we can see the different peaks obtained by the different compounds of Pseudomonas fluorescens. This is the structure of the DL-leucinamide N2N2 dimethyl. And this extract of Trichoderma asperellum and Pseudomonas fluorescens were further studied against the pathogen by using poison food technique at 3 different concentrations. And at all the concentration, there is a reduction in the growth of the pathogen, and at 15%, that is maximum concentration, the maximum inhibition of the pathogen observed. Hence, it is proved that the volatile constituents of both Trichoderma and Pseudomonas were inhibitory to the growth of the pathogen. Here we can see the inhibition of the pathogen at different concentrations. Coming to my third objective, compatibility of efficient isolates of native Trichoderma and fluorescent Pseudomonads with recommended fungicides. The fungicides were, the compatibility was tested by poison food technique, in which the percent inhibition of mycelial growth of the Trichoderma were recorded, and compatibility was recorded based upon the inhibition. Among the systemic fungicides used at 3 different concentrations, azoxystrobin alone was compatible with the Trichoderma asperellum. Here we can see the maximum growth of the Trichoderma asperellum in azoxystrobin treatment compared to all other treatments. In case of non-systemic fungicides, propinep and copper hydroxide were compatible with the Trichoderma aspergillum with no mycelial inhibition. Here we can see the complete growth of the Trichoderma aspergillum as compared to the other treatments. And coming to combi fungicides, in which copper oxychloride plus copper hydroxide and cymoxanil plus mancozeb were highly compatible with the Trichoderma aspergillum, which allowed the complete growth of the Trichoderma in the treatment. In case of Pseudomonas fluorescens, the modified paper disc method was used and zone of inhibition was recorded, in which the azoxystrobin, hexaconazole and tebuconazole were compatible with the Pseudomonas fluorescens with no zone of inhibition was formed. In case of non-systemic fungicides, propinep alone was compatible with the Pseudomonas fluorescens with no zone of inhibition. Here we can see there is no zone of inhibition. In case of combi fungicides, thiophanate methyl plus pyroclostrobin and cymoxanil plus mancozeb were compatible with the Pseudomonas fluorescens. These are the list of chemicals which are compatible with the Trichoderma as well as the Pseudomonas fluorescens, and the cymoxanil plus mancozeb combi fungicide, which is compatible to both isolates, was further used for the management of wilt of chickpea. In management of wilt of chickpea under glasshouse condition, one efficient Trichoderma isolate, Trichoderma aspergillum, and Pseudomonas fluorescens were used at consortium, that is stock-based formulation was used in the consortium, and this cymoxanil plus mancozeb chemical was used, in which there is a less disease incidence in the treatment, that is seed treatment with cymoxanil plus mancozeb, followed by soil drenching with the same chemical, followed by the seed treatment with the talc-based consortium of Trichoderma aspergillum and Pseudomonas fluorescens, followed by soil drenching with the cymoxanil plus mancozeb. Here we can see the less disease incidence in both the treatments as compared to the control. In conclusion, 20 native isolates of Trichoderma and fluorescent Pseudomonas, they showed antagonistic activity against the pathogen, and Trichoderma 14 and PF19 have shown maximum inhibition. Again, 20 isolates of Trichoderma and fluorescent Pseudomonas, they produced volatile compounds which inhibited the growth of the pathogen with higher concentration of compounds produced by TR14 and PF19. 29 compounds were obtained in extract of Trichoderma aspergillum upon the GCMS-MS analysis with high concentration of 2-imidazole 1,9-methyl pyridine 1-oxide with a retention time of 19.842. And 55 compounds were obtained in extract of Pseudomonas fluorescens upon GCMS analysis with the higher concentration of compound DL-leucinamide N2N2 dimethyl with a retention time of 7.923. In case of chemicals, azoxystrobin, propinep, copper hydroxide, copper oxychloride plus copper hydroxide, and cymoxanil plus mancozeb were compatible with the Trichoderma aspergillum, whereas azoxystrobin, hexaconazole, tebuconazole, propinep, thiophanate methyl plus pyroclostrobin, and cymoxanil plus mancozeb were compatible with Pseudomonas fluorescens. In case of pot culture experiment, the seed treatment with cymoxanil plus mancozeb followed by chemical drenching with the same chemical around the infected plant is effective in reducing the disease as compared to the control. Thank you, everyone.
Madam, unmute and talk, please.
Thank you very much, Dr. Ranjana, and very exhaustive work on different isolates of Trichoderma and Pseudomonas. And you also have found out an effective formulation. And I hope that you have taken care of proper identification of Pseudomonas, because of late it is becoming very tricky.
Yes,
and so you should use maybe different loci to find out the correct identity of the isolates. Anyway, congratulations, and go ahead with your work. Okay.
Thank you, ma'am. Thank you, ma'am.
And timing is 12 minutes, 55 seconds.
Okay.
Almost 13 minutes. Okay. So now I call upon Jayalakshmi.
Ma'am, am I audible, ma'am?
Ranjana, please stop sharing.
But you have to share your screen, no?
Yes, ma'am.
And check it up whether your PowerPoint is coming properly and all.
Jayalakshmi, on your video also.
Ma'am?
Video.
Video. Your face is not
Face
visible.
is not seen.
Okay, ma'am.
Is it okay? We are not able to see you.
Yes, ma'am. Wait for a minute, ma'am.
Click on video. It is muted. Yes.
And put your PPT in full screen.
Shall I start, ma'am?
Yeah.
Not clear. Jayalakshmi, 1 minute.
Shall I start, ma'am?
Okay. Now it's
Good afternoon to everyone. My topic is studies on gliotoxin and gliotoxin producing trichoderma virens on the management of tomato damping of disease. First, the gliotoxin is a sulfur-containing antibiotic, which is first originally discovered from Gliocladium virens, and it is toxic against various soil-borne pathogens. Hence, it was called as gliotoxin, and it has broad spectrum of activity against antibacterial and microbial activity, and it is mainly due to the presence of disulfide bridge within its molecule. And apart from Trichoderma virens, other genus like Aspergillus fumigatus and Neosertoria pseudofisherii and Penicillium species and Acrimonium also producing gliotoxin. And all the Trichoderma species do not produce gliotoxin. Except Trichoderma virens. In Trichoderma virens itself, certain strains called Q strains only produce gliotoxin, whereas P strains produce only gliovirin. And in Trichoderma virens, the antibiotic production is an important triad by which it effectively suppresses the soil-borne pathogens. And first time the Trichoderma virens strain 20 was commercialized as a biopesticide in the name of Gliocord. It produces gliotoxin enormously, and the gliotoxin production is directly positively correlated with the suppression of plant diseases. And the 13 gene cluster was in 13 gene cluster was governed for GT biosynthesis, and recently the Bulgari 2020 they identified the GlyH, a protein for GT biosynthesis, and it is not present in a non-producer of gliotoxin. And my study has three very few studies on related to gliotoxin and gliotoxin producing Trichoderma virens strains, and there is no information regarding about accumulation, persistence, and dissipation of gliotoxin in soil. And we should have some knowledge on factors influencing gliotoxin, stability in soil also. And keeping all these points in mind, my research work was having following objectives. First one is isolation of gliotoxin producing Trichoderma virens strains, and second, analysis on its persistence in plant and soil, and third one, effect of gliotoxin against various soil-borne diseases in tomato. First, the rhizospheric soil was collected from various parts, and they were undergone serial dilution, and they were identified under microscopic observation. The main difference of Trichoderma virens from the other Trichoderma species is it produces Penicillium type of conidiophore, in which the phyllates are produced at the end of the conidiophore without secondary branching in acute angle, and whereas here we can see the well branching, and the phyllates are in a right angle manner. And to differentiate the P strain from the Q strain, the TSM was amended with 50 microgram of gliotoxin, and which allows the only gliotoxin producing Trichoderma virens, and that was alone further inoculated into the veining medium for gliotoxin production, and they were extracted with chloroform and further conformed with the TLC, thin layer chromatography method. And by this way, we have isolated 16 Q strain and 10 P strains. And all the 16 Q strains were tested on gliotoxin production. Among the isolates tested, 16.1 shows the higher production of gliotoxin, and this was further used for further studies. And the gliotoxin was large scale purification of gliotoxin was carried out with the help of preparatory TLC plate, and further it was and confirmed with the HPLC analysis, and both in standard as well as in sample, the peak was elicited at 3.3, which confirmed the presence of gliotoxin. And the gliotoxin was production was started at second day, and it extend up to 5th day, and after that it it started to convert it into the another compound, and when time extends, it conversions also get started into the to form a other compound that is called modified gliotoxin. And this was further confirmed with the HPLC analysis. Here on third day, we can see only gliotoxin peak. On fifth and 10th day, due to the conversion, that is called modified gliotoxin. And that modified gliotoxin was identified as methyl thiogliotoxin by LCMS method. And coming to second objective, there is no information on extraction efficacy study on gliotoxin plant and soil ecosystem, for which the purified gliotoxin was artificially spiked in soil, and different solvents varying its polarity was used for gliotoxin extraction. Among the solvents tested, the chloroform um, showed the maximum extraction efficiency of gliotoxin from soil. Likewise, in coir pith and FIM and vermicompost, in all the three organic manures also, the chloroform showed the highest amount of gliotoxin extraction. And in plant also, ethyl acetate extracted the gliotoxin in an effective manner. And we should also have the knowledge on factors influencing stability of gliotoxin in soil. The first factor is pH. The acidic to neutral pH favors the gliotoxin stability even up to the 1 month of period. With regard to temperature, compared to the 30 and 50, 80 degrees Celsius degrade the gliotoxin very fast, and less than 50 degrees Celsius will enhance the gliotoxin stability for longer period. And with regard to light, complete light as well as alternate light and dark favors the gliotoxin stability in soil even for 30 days. And with regard to moisture, the wet condition inhibit the gliotoxin stability. This might be due to the presence of microbes which degrade the produced gliotoxin. And finally, the analysis of gliotoxin on the management of damping of disease of tomato was analyzed. And here the pythium was isolated from the infected tomato seedlings, and they were identified as a morphology character as well as molecular level. It was confirmed as a pythium acynodermatum. And the purified gliotoxin as well as modified gliotoxin was tested against mycelial growth of pythium, and the gliotoxin showed antifungal activity at 50 ppm, and whereas the modified gliotoxin amended plates does not show any such kind of restriction even at 150 ppm. And upon spraying of gliotoxin directly on tomato leaves, it showed some scorching effect on 750 ppm as well as 1000 ppm. But upon seed treatment with gliotoxin for overnight, there is no significant difference was observed at the shoot and root length from treatments between treatments and control. And finally, the culture filtrate of Q strains were tested against the damping of disease incidence, in which the culture 100 ml of culture filtrate applied on the soil at 4 different time period greatly suppressed the damping of incidence under in vitro condition. And coming to the conclusion, except Trichoderma virus, none of the Trichoderma species produce gliotoxin, and the modified gliotoxin was identified as a dimethyl thiogliotoxin by LCMS method. And for effective extraction study, chloroform and ethyl acetate found to be very useful for extraction of gliotoxin from soil as well as in plant. And the factors like acidic pH and low temperature and alternate light and dark and moisture condition, low moisture condition favors this GT stability, as well as GT has no phytotoxicity effect on tomato plant, and seed treatment with the gliotoxin effectively suppress the soil-borne diseases in tomato. And coming to future prospect, this studies greatly elaborate the knowledge about gliotoxin for crop protection manner. Thank you.
Thank you very much, Dr. Jayalakshmi, for your very precise account on gliotoxin and how it can be extracted and how it is useful in preventing the fungal diseases. So, thank you very much. But it is not acidic, but it is acidic. Okay.
Okay.
Acetic acid.
Timing.
But the acidic is not T but D. Take care. Okay.
Okay. Okay.
Timing 8 minutes. Madam, 1 minute. Timing 8 minutes 39 seconds.
Okay. Next, I.
Madam, that yes, Gayatri is there.
But is she ready? Has she loaded?
Yes,
her
I think she's
PPT?
there. Gayatri.
Ma'am, yes, ma'am.
Please load your PowerPoint, check your audio, video, everything, and tell me. Then we will start.
Okay, ma'am. Ma'am, whether my slide is visible?
Yeah,
Yes.
slide is visible, but you should also be visible.
Okay, ma'am.
On your video, Gayatri.
Okay,
Your
ma'am.
face is not seen.
Okay, ma'am. Ma'am,
There's no light. You are looking dark.
Sit opposite to light.
You sit opposite to light source, yeah? Why you are sitting?
ma'am.
Yes.
Now it's so. Again you are sitting.
Okay.
Sorry, ma'am. Ma'am, shall I start, ma'am?
Yes.
Good afternoon to
1
all.
minute, 1 minute.
Okay, ma'am.
Now you can start.
Okay. Good afternoon to all the delegates of plant pathology. Myself, M. Gayatri, MSc Plant Pathology from Tamil Nadu Agriculture University. I'm here to present my research findings on elucidating the antifungal biomolecules of Canoderma lucidum against mango anthracnose pathogen Polytotrichum gliosporioides.
Next.
Okay. The presentation will going to cover the following headings, which include start off with introduction, then need of the study, and the work carried out under the research work and their result, and finally conclusion. So let's get started with an introduction. Mango is one of the major consumed and reputable fruit in India, and it also affectionately called as king of fruit because of its high nutritive value, richness in vitamins, and rare sugar content. Such a most important crop is being affected by various pests and diseases. Among those, mango anthracnose is one of the major devastating diseases due to its pre as well as post-harvest infection, which can causes an economic losses of even up to 100% based on prevailing wet weather humid condition. So far, several fungicides have been registered for the management of the disease, which ultimately result in health hazards, environmental pollution, and also resistance in pathogen. Moreover, the export potential of mango was further affected by pesticide residues. So here comes the need of developing fungicide formulation from various natural sources might provide way to tackle the problem. And also developing fungicide formulation from biomolecules of mushrooms like Canoderma lucidum act as a promising alternative to chemical fungicide and added advantages of minimum mammalian toxicity and also eco-friendly measures, which will ultimately result in pesticide-free crop produce. So let's move on to the work carried under the study. So initially, we have started with extraction of secondary metabolite from mycelium of Canoderma lucidum. For that, the mycelium of Canoderma lucidum was inoculated into the PD broth, and after 20 days, the culture filtrate was collected from the broth, and here we have chosen ethyl acetate as a solvent for extraction of secondary metabolite. So the equal volume of ethyl acetate was added into the PD broth, and then the flask was kept in shaker for overnight, and then the solvent fraction was evaporated using vacuum plus evaporated, and then final residue obtained was dried and then scrapped using methanol. So the final solution obtained from scrapping is used as a stock solution for further antimicrobial analysis. So here further the efficacy of biomolecules collected from the mycelium of Canoderma lucidum was tested against Polytotrichum gliosporioides through agar-well diffusion. The result showed that the maximum inhibition of 68.55% was observed at 1% concentration. Here, have a look at this. Here, the picture shows the maximum mycelial inhibition of 1% at maximum mycelial inhibition of 68.55% at 1% concentration. This clearly shows that the inhibition is due to the presence of certain compound that would be responsible for the retardation of the growth of the pathogen. So, hence the characterization of this biomolecule is necessary for the identification of those compounds. So, GCMS analysis was carried out. The result revealed the presence of several certain compounds like formic acid, pentaethylene glycol, hexadecanoic acid, papaverine, etc. Even though the several compounds were identified, among them, the compound papaverine has retained its higher probability area with higher peak area percentage. Further, FTR analysis was carried out to know their functional group of the compound. The spectrum in the picture indicated the presence of several groups like aliphatic primary amine, amine salt, thiol, amide, ketonic groups. So it further indicated that there was a presence of certain biomolecules, certain compounds in the biomolecules of mycelial culture filtrate of Canoderma lucidum. Further, TLC studies were carried out by using a mobile phase of chloroform is to methanol is to water at a proportion of 30:4:1, indicated the presence of two bright bands under UV light. Further, the obtained bands were scrapped and their antifungal efficacy was tested through agar-well diffusion. It showed an inhibition of 36.6% in band 1 and 40.5% in band 2. However, the compound papaverine is obtained from GCMS analysis as retained its high peak area percentage. Our portion of literature also shows that the compound papaverine had a strong antiviral and antimicrobial property against human pathogen. So far, it is not exploited in case of fungal plant pathogen. So, the study is intended to test the standard sample of papaverine against the mycelial growth of Polytotrichum gliosporidis through agar-well diffusion assay. It shows the compound papaverine was required a minimum concentration of 1500 ppm was sufficient for 100% inhibition of mycelial growth of Polytotrichum gliosporidis. Further, it was cross-checked with the paper disc assay. Here also we found that 100% inhibition in 3500 ppm. The graduation in the concentration level between the agar-well diffusion and paper disc assay is due to the variation in the rate of infusion level and also evaporation level. That's why the higher concentration was required in case of paper disc assay. Further, the conidia is a major character for the this is infection. So that the spore germination assay was further carried out in papaverine. It shows in 24 hours of incubation, the conidia gets germinated. Even though the conidia gets germinated, but the spore inhibition was reduced to 50%. So it clearly shows that the compound papaverine had both antifungal as well as spore germination inhibition activity. So let's move on to the fruiting body of Canoderma lucidum. Partial of literature shows that the fruiting body of Canoderma lucidum have copious antimicrobial compound. So that the fruiting body of Canoderma lucidum were cultivated at Department of Plant Pathology, Tamil Nadu Agriculture University, under the guidelines of DMR Solan. So, and the cap and stripe portion were harvested and it is stored for further studies. In case of fruiting body, initially the biomolecules from cap and stripe portion were extracted separately. For that, the cap and stripe portion was ground into a powder, and then for 10 gram of a powder, 100 ml of ethyl acetate were added, and then it was kept in incubator for an kept in a shaker for overnight, and then using vacuum flask evaporator, the solvent fraction get evaporated, and then finally the residue was dried and scrapped using methanol, and it served as a stock solution for further analysis. So hence further the antagonistic efficacy of biomolecules from cap portion of the Canoderma lucidum was tested against Polytotrichum gliosporidis. It shows a maximum inhibition of 70% at 1% concentration. Have a look at this picture. It shows the inhibition of 70% at 1% concentration. And the style portion, similarly, the style portion was further analyzed against the mycelial growth. It shows the inhibition ranging from 34 to 40 at a maximum inhibition was obtained at 1% concentration. The picture shows the inhibition percentage of 40.77 in case of 1% concentration. So coming to the result, comparatively, cap ocean possess more antifungal activity against the tested pathogen compared to style. So the cap ocean was further subjected to future studies. So the characterization of the compound is much more needed to identify the antifungal compound. Hence, it can be done through GCMS analysis. Here also, we can find several compounds like disoxyl, like docosahexaenic acid, propionic acid, and benzothiazole. Among those, benzothiazole has retained a higher probability percentage with high peak area. So it was used for further studies. Further, it was confirmed through TLC analysis. Here, we have obtained 2 banks, and the obtained banks was further scrapped, and it was tested against the pathogen. It showed the inhibition ranging from 46.3% in case of band 1 and 53.7% in case of band 2. So the compound benzothiazole was obtained from GCMS analysis. The partial of literature shows that the benzothiazole had a strong and possess antifungal and antimicrobial property, and also similar studies has been reported, like it inhibits the growth of Phytophthora capsicum through inducing apoptosis and cell membrane. So the compound has been purchased from Sigma Aldrich, and their efficacy against the pathogen was tested through agarwal diffusion assay. Here, the result obtained showed that very minimum concentration of 50 ppm was more sufficient for complete inhibition of the pathogen, and also the growth pattern of the mycelium was observed. It showed that the mycelium pushed back and distortion of mycelium and also blackening of mycelium in case of treated plates. And also, it was further cross-checked with the paper disc assay. Here also we have found that at a concentration of 75 ppm, it shows 100% inhibition. And also some growth changes in the plate was observed in case of paper disc assay too. It shows the linear mycelial growth pattern towards the paper disc, and also the shrinkage of mycelium in 75 ppm. So further, the spore germination assay was carried out. Here, the benzothiazole completely inhibit the germination of the spores even at 12 and 24 hours of incubation. This shows that the benzothiazole compound possesses both antifungal as well as spore germination inhibition activity. But even compared to pepaverine, benzothiazole performed well because of 100% inhibition in both mycelium and spore germination assay. So the effect of benzothiazole on the mycelium and spores of Polytrichum gliosporides was observed under some studies. It shows a clumping of mycelium and distorted growth in case of the treatment, and also misshapen conidia and also ungerminated conidia, whereas control plate shows a normal mycelium growth pattern and normal shaped conidia, and also germination of conidia was observed in case of control. So coming to a conclusion, the present study overwhelmingly indicated that the presence of several innate and novel biomolecules in the fruiting body of Canoderma lucidum, and it was performed well in inhibiting both vegetative and reproductive structures of Polytrichum gliosporides, as substantiated by the identification of several antimicrobial compounds through GCMS, FTIR, and TLC studies. Thus, Canoderma lucidum provides spectrum of opportunity for developing eco-friendly measures for management of anthracnose disease. I would like to express my special gratitude to my chairman, advisory committee members, and professor and head coordinator, Department of Plant Pathology, for providing mental guidance for completing this study. Thank you. Thank you for your attention.
Thank you very much, Dr. Gayatri. It was very, very novel type of work, like trying to find out a biomolecule from the mushrooms.
I'm just completing my MSc degree now.
Okay, no, you will definitely join for PhD. My best wishes to you.
Thank you so much, ma'am. Thank you.
Okay.
Madam, 12 minutes 48 seconds.
Sorry, ma'am.
Okay, now how do we go about it? Any suggestion from anybody? Like,
Madam, here we have had 5 presentations, and but as per the given schedule, Dr. Nanatambi has provided only MJN Narasimhan Award Contest, but we asked the organizer for 2 awards. One is MJN Narasimhan Award, second one is APS Travel Grant.
Yeah.
And MJN Narasimhan Award, that both MSc completed and also doing PhD, or this MSc PhD students can are eligible, and for APS Travel Grant only PhD students are eligible.
Agreed.
And for MJ Narasimhan Award, only one name should be is recommended for final contest.
No, 2.
Two
MJ Narasimhan Award, two people. Both for 2. 2 each.
No, but the other one, that APS award, we have not got. Now you will be announcing, Dr. Nalatambi?
No, ma'am, there is no contestant for that award. I requested all the campus.
No, but out of these 5, can we select 2 names?
No.
APS?
No, Raju, that is for the
postdoctoral
PhD. This is
PhD. Yeah.
No, here I have seen.
Madam, for the kind information, of course, I should not tell this. Many students are scared about the COVID. They told me that, sir, we don't want to travel.
No. One thing, no, I would like to clarify here.
Last time,
so here, you see, for the benefit of the students, if you can recommend 2 names each for both the awards, that will be benefit for the students. And those who are pursuing PhD, maybe 2nd year or 3rd year PhD, they are actually eligible for APS Travel Grant. And it is not like that. Suppose those who will be finally getting that APS award, this Travel Grant award. So under this situation, this year, what has happened in the last August month, when American Phytopathological Society organized this international symposia. So 3 candidates were qualified from IPS, and all the 3 got the chance for virtual presentation, because there is no question of traveling. We don't know in the coming year also whether the situation will be improved or not. So in that case, they can take the advantage of this virtual presentation, because they won't get the chance of traveling.
Even registration also.
Yeah. In that case, the secretary agrees that we can, once again, you can issue a circular for all the camps to apply the hard copies, and we will scrutinize and forward to you if you don't want the presentation.
And actually, no, presentation means finally from 8 zones, they are asking, APS is asking to give them 16
2 names from each.
Yeah, each zone. So out of the total 16 candidates, maybe less than 16. So they will be giving this that award to 2 students. We don't know.
Yeah, Dr. Robin, I fully
that's
agree.
why if you can recommend 2 names under the category of APS Travel Grant, that students will be getting the benefit.
That is true, absolutely true. But the thing is that, can you give some more time to do this job?
Yes.
Because our zonal meeting ends today. If you give some more time, maybe another couple of weeks or so, we will take our possible efforts to send the circular to all the campus in the South Zone, and we'll invite the application. We will say same way we'll put a panel to scrutinize the application, and we will forward to the headquarters. Then you
yes, okay.
Yes, sir, that can be done. That can be done.
Okay, then formally I'll request you kindly give me consent. I'll immediately take the action. Okay.
Actually, for the favor of the students, actually we are telling.
That's what. It's only for PhDs for travel grant.
Nalatambi already they have given the approval. Now you can go ahead. You need not to look.
I want black and white, sir.
What black and white? They are accepting.
No WhatsApp message.
When secretary is accepting, what's the problem?
2021 will come.
There are so many.
It is very well black and white because it is recorded. You can claim it.
Yes.
Yeah, that's
Yes,
right.
yes.
yeah.
That's true, sir.
But
Because other zone should not claim, na. Dr. Nalathambi took extra time and he is recommend the candidates.
She wants to make some comment. Malti madam.
No, I just want to know, like, we will come to a conclusion and then get back to you. Like, we can come out of the meeting and do it, or how do we do
yes,
it?
yes, yeah.
Madam, you can
take your
come out
time.
of the meeting. Kindly communicate to me that the mail which I have sent to you.
Okay.
Kindly discuss with all your members and the jury and give the list, and then I'll put it the final validity function. That's all.
Okay.
And Dr. Nalathambi,
yes, sir.
I request you, those candidates who have already participated, you have to ensure whether they are member of IPS or not.
Which participation?
They first thing they
that certificate we received. Yeah, after we have seen the certificate, then only we allowed for the presentation. It is there.
Okay. But they have registered as a member.
Yeah. Already they are life member. Some are the annual members. And their certificate they received from the respective chairman for the work. So all these formalities we completed.
Okay.
It will forward to you along with that recommendation.
No, it's okay. Then you take your time, and judges and all panel members also can take time.
Yes.
You decide who will be put in the category of Narasimhan Award final contest in the annual meet of IPS, and proposal for this APS travel grant. 2 names.
No, proposal APS grant in this panel it's not possible. I personally.
Yes, okay. You keep pending. You take time.
Yeah, we take time.
We have conducted for MJ Narasimhan. That is over. Only for APS grant, it will be recalled again.
Yes.
That recommendation should come before our annual meeting.
No, that is true, Kalyan, but because we didn't receive any application for that travel.
No problem. In the meantime, you have time. So you can just process.
December last week will be conducted. No problem at all.
No, but we cannot conduct this presentation. Sir, one thing I want to clarify, Dr. Robin.
Yes.
For AP suspend the travel grant, we cannot conduct a meeting like this. We will scrutinize only the hard copy of their application with the panel of members.
No, you can conduct it. No, you can conduct it. Let them present if you want. What is
with the panel, you can conduct it. Because without presentation, it cannot be decided.
You conduct it.
See, like this, we can have it. Even whether other person also can join, there is absolutely no problem. Only link will be provided by headquarters, which these people will be presenting with 2 hours time or something.
Yes. Then okay, Dr. Magesh, why don't you merge this thing to the other zone, which subsequently they are going to
here, so
it will not be done. See,
so Dr.
You see, Dr. Robin,
yes,
kindly listen to me. The practical difficulties. You see, with a lot of difficulties, we could manage this meeting, and we could bring all the people for today. Then again, doing the same exercise for one award presentation, I think it will be cumbersome for me. So what I would suggest, whether we will receive the application, we will request the candidate to present the same thing on behalf of the South Zone in the forthcoming zonal symposia. There are 7 zonal symposia.
No.
Other symposia.
Don't club.
No, don't. If you have problem, I will conduct it. There is no problem at all.
What you are bothering? I don't know.
No problem at all. I will conduct on behalf of you.
No. You see,
see,
not like that, Magesh.
Our zone, it should not be overlapped with other zone.
Then how do you bring all this? Can you make again registration link and so many things?
No.
IPS will do it.
Dr. Nalathambi, just let me clarify you. One thing, so I must give you a very big thank you to you, because within a very short notice, you could organize this meeting, because you are the first, your zone is the first to hold this zonal meeting within 15-17 days of our decision.
Yes.
So with that capacity, I can give some extra time only for holding the APS travel grant competitions among the students. So that you should complete, suppose within 1 month or before
annual symposium.
Before annual meet, because our zonal meet will be ending. Last one is 27 and 28 of January, and that you take your time. That will be hardly 2 hours matter.
That is true. Whether Dr. Robin, in that case, whether you will be able to give the free link to all the participants?
Yes, we can give, sir. No problem.
Okay, okay, fine.
You can give, sir.
So the registration may not be the mandatory.
Nothing. Nothing.
Okay.
Only
So these
mandatory,
two.
they should be, the participant should be member.
Yes.
Dr. Magesh.
Only
non-member
presenting
also can
presenter will be member.
Yeah,
Oh,
presenter
no.
only.
Presenter should be member.
Presenter should be member, even, but the audience can be even non-member also can participate for the
Yes,
presentation.
yes.
No.
Yes.
You cannot restrict only the members.
This is only presentation, only no question answer session. So there is.
That is true, man. You try to understand, but the audience can
Anyone,
see
no
the
problem.
presentations.
That
You cannot restrict to only our members.
that you will be getting the link, which like this, you have the now list. With that mail ID, you circulate it. If they want,
Yes,
they can find, there is no
Yeah,
problem.
that we can do. That we can.
That's what. That is the only work is there.
That can be done within a week also.
Within a week also you can do. Within
Guruji, you Dr. Magesh, you promote 5 students from your state. I request Dr. Magesh to promote 5 students from the Kerala state. I am ready to conduct the meeting.
Okay, we will explore it.
Yeah, sure.
Yes. No, it's good.
Only
Because
consider if they, whoever comes, we will try.
Sir, we have advantage of this actually virtual system, na?
yeah, we are giving that much of
We'll provide the
benefit
link
to you.
We'll provide the link and it can be conducted. No problem.
Yeah, okay, we will conduct it. May you give me some more time, another 15 days time. 15 to 20
Yes,
days.
yeah.
Okay.
Within this year, you finish within this year.
Okay, sure. Within this year means
2020, sir.
Okay. Not taking much time. We will allow the panel to finalize the list, and because we have 4 o'clock the validity function.
Yes. Yeah.
Thank you, madam. Thank you, sir. Dr. Velavan, sir, all the way from Muscat, you joined with us. It's a great pleasure for us, and all the judges from timely conducting the session. I am really thankful to all of you, and particularly senior persons like Dr. Sridhar and all people, and kindly all of you join the validity function. That's in fact for all the seniors and a few allied people, I have sent the personal letter also today early in the morning around 2 o'clock. They kindly make it convenient part of the validity function, and perhaps the registrar from TNA has kindly consulted for the chief guest. And thank you very much, sir. We will join sharply by 4 o'clock.
4 o'clock, na? Yeah.
Yeah, Robin. Robin, anything want to convey?
No, from this panel member, let's hear who are there. 1 minute. Is there? If they are having some.
No, they'll interact with you. It's a common. How can you disclose these are the churches, this and that?
Yeah.
Madam is there. She will take care.
Okay.
Okay.
Yeah.
So see you after 15 minutes, sir. 15